Well, this is part 7 of our discussion of cytology. This is the last lecture part on this. And we're back into discussing the changes in chromosome structure. This one's going to be fairly complicated, so you may need to watch this one a couple of times to get all the nuances of it, but we'll try to make it as simple as possible because the rearrangements and certainly their outcomes are very important. So we have rearrangements. We're going to talk about two kinds. We're going to talk about inversions, okay, and we're going to talk about translocations. Now remember, once again, we're in chromosome structural changes, so we're not going to gain or lose amounts of DNA, at least from a genomic standpoint. Okay, when we go to translocations, we're going to lose it potentially from a particular chromosome, but it's going to be somewhere else in the genome. In an inversion, we don't leave the same chromosome. We just changed around the order of things in the chromosome itself. So let's start by looking at inversions. Okay, inversion is just what I said. It's where we're going to have our little stylized chromosome here, A, B, centromere, C, D, E, F, whatever, and we simply invert some arrangement in the middle. Okay, so we might take that and have it change over to something where we have A, B, C, F, E, D. Okay, that's known as an inversion. Inversions can be relatively minor in the sense of their outcomes. If all the DNA is there and nothing has been a problem with the genes, then it's okay. If not, it can be extremely severe because let's say that C has the control region, is part of the control region for the D and E genes, maybe, or something like that. Well, now if those are moved completely away, where C is now facing F, there's no way to adequately control D and E or any of the others. So you may end up with fairly significant problems if you have these types of inversions. So they can range from basically undetectable except by karyotyping, or they can range to highly, in fact, all the way to lethality if you have them in the regular systems. So it just depends, okay? All right, well, how might we get this? Why would DNA just flip around? Well, we go back to the same arguments that we've made before. In this particular case, we said, all right, if we think about our DNA and it's flexible and things that can loop around, and one of the things it can do is loop. If you have a break that occurs in here somewhere, okay, you can have lots of things happening. You could have a formation like this, which would give you a link or a ring chromosome or something, and an internal deletion, or you could have a situation where you simply fuse it back, okay, but you fuse it back incorrectly. So you fuse it back looking something like this, and if you do that, then you're going to end up with what we see over here, and not right. Back, there we go. You'll end up with something where you have this nice little component looks something like this and like this. Now you notice that will be an inversion, right? That's what that would end up providing for us. Okay. All right, so if you have those, you have two possible types. You have what's known as a paracentric inversion, something like this, okay? Paracentric inversion. Para means it's near or around, but it doesn't involve directly around the centromere, okay? Like a paralegal or a paramedic is someone who's not fully an MD or someone who's not a full lawyer, but they have basic things, so they're near, close to that. Well, so in this case, we're para, P-A-R-A, we're not going to involve the centromere in this case, and so we're going to flip around. And notice we got A, B, centromere E, D, C, and F. Now what's important here is we didn't change the arm length. As you go back and think about some of that early stuff we asked you about chromosomes and P and Q arms and long and short arms, one of the things that happens is that you go from a metacentric to a submetacentric or vice versa, but here you don't. The chromosome would appear the same as far as the length and location of the centromere, right? Now the juxtaposition of this, we would have a chromosome where we have a pericentric, peri like periscope, right? Periscope is classical scope in submarines where you can look all the way around, okay? So the para means you can go around, and para in this case means that you are around the centromere. So the breaks that would occur would be somewhere on both sides of the centromere. Now what happens in these cases is usually these breaks are not equal, right? So what will happen is that you'll get more material on one side of the centromere than you will on the other. And when that happens, when you invert it, you end up changing the position, the relative position of the centromere. So in this little example here, we had a submetacentric, right? So if we were to stretch this out, A and B out here, you'd see that the centromere is up closer to the top than it is exactly in the middle. But if we flip that part, now it becomes relatively close to a metacentric chromosome. So we would move the centromere down by simply doing that flip. But once again, it's an inversion. You have A, D, C now instead of A, B centromere. So you have this part that's been moved around. Okay. Just some examples of where this has been important. I showed you before some relationships between human chromosomes and some of the great apes of chimpanzees, and showed you where we have many of the same genes being shared. Well, one of them is on chromosome 4. On humans, the difference between humans and chimps is that humans have had this pericentric, notice it's peri, it's around the centromere, inversion. Okay, so if we look at the chimps, they have this form that looks like this. In fact, it's common in the great apes. And we get the humans, we've had a nice flip. So it's just flipped that part around in chromosome 4. But the same basic information, the same genes are actually found in that region on both humans and chimpanzees. So this all works fine, excuse me, until you end up with trying, if you're going through mitosis or whatever, again, it's a position effect. If the what's been flipped is what matters. If it's been flipped in a region where it could cut and move and there's no change, then your genes are probably working, everything's fine. If it's a situation where you've interrupted a control or you've split in the middle of a gene or something else, then obviously you're going to have much more problematic. But ultimately, where it's going to have trouble is when you go to synapse, when you go into prophase. When you try to line up these sister chromatids, replication doesn't care that they're inverted, right? Replication says, I don't care if this is inverted, I'm going to make a perfect copy, all right? But what happens is when they try to line up, they run into difficulty because this end and this end, right? They have to match, they have to flip. So what'll happen is they'll try to line up with A and B and centromeres, and then they start to have difficulties, okay? And those difficulties, well, sorry, trying to get my pen back. Those difficulties, remember when we had before, we showed you that with deletions or things, we'd have one of them out like this, and then the other one would come along and bow out like that, right? Well, you can't bow this out. You can't, there's C here, has to be connected up with C up here in some way, right? And of course, D in this case is fine, and E has to be up here. So you can't just draw a line through there and say they're going to connect like that. Somehow, physically, this piece of DNA has to be connected to this piece of DNA off down here, right? Well, the way they're going to do that is through what's known as an inversion loop. And this is what you would actually see when you go into meiosis, and if you had got late prophase and you went into late prophase, you would actually see these types of inversion loops like this, okay? These inversion loops work out fine. It now puts C with C and D with D and E with E, and everything looks like it's going to work out just fine, okay? The problem, of course, is that we know now that in most cases, we're going to have crossover somewhere, right? Well, if you have crossover inside of this inversion loop, it can end up giving you all sorts of problems, okay? And this is where the book illustrates it right here. Okay, so again, the crossover could be anywhere. I mean, in any one of these chromatids, but we're going to put it here. You notice that the outer one, we'll just pretend the outer one doesn't cross over. You have A, B, centromere C, D, E, F, G. Everything's fine out there. Okay. Same thing down here. If we follow this third one around, it's actually kind of weird, but if you follow, you got A, B, C, centromere, internal loop, nothing really happens. It's just an inverted chromatid. Okay. But look what happens with the crossover. If there's an odd number of crossovers, okay, within the inversion loop, you get a really bad outcome. If there's an even number, you cross over and you cross right back, it's not as problematic. But if you have an odd number, like 1 or 3 or 5, in this case 1, look what happens. You have A, B, centromere, and you should probably try to print this out and do it with your pencil, pen. C, okay, we cross over, we pick up D. Now we've got E, but we're starting to go back the wrong way, right? We come back this way, we pick up another centromere and another B, and another A. So we have 2 A's and 2 B's and 2 centromeres, and it really becomes messed up. And then, of course, if you go the other way, right, if you think about what's happening in the other direction, well, wait, they're not connected at all with the centromere anymore. You have to start on this end and say I've got a G, an F, looks like I've got an E and a D. I cross over and pick up a C, but then I start going back the wrong direction again, and now I got another E and F. But this one doesn't have any centromeres, right? You follow that around with your pencil, there's no centromere, right? So you end up with a very problematic outcome here. If we have this crossover and we start to pull this all apart, obviously with the anaphase 1, right? You're going to have difficulty. For one thing, this is going to be interconnected by two centromeres. This is not going to have a centromere at all. And then you're going to have all these kinds of problems. So what you tend to end up with is this thing called a dicentric bridge on one side. And then you end up with this chromatid without any type of centromere. So it just wanders along in some place or the other, kind of lost, right? Now, if you're strongly pulling these apart, you'll often break this piece of DNA somewhere, this chromatid, or if you don't, you'll end up with actually a dicentric. Now, the book shows it as breaking, and that's fine. What it's showing here is you get, on the outside, there's no crossover, so you get a normal chromatid. On this side, and this one down here, number 4, you have them inverted, but you didn't have any crossover, so you just end up with another inverted chromatid. So we've got this one and this one taken care of. All right. This one is generally lost because it doesn't even have a centromere. Okay, we call it acentric, and we'll do that in a moment. And then they're showing this one is broken. Okay, and so you come along, you break it maybe somewhere in here, and you pick up D and E, but you break it, and you do that with 2 chromosomes that look like this, both having centromeres, but both being major deletions. And this one even being an odd one because you've got this inversion on your deletion. And of course, if these become the chromosomes that are used to make the sperm or the egg or the ova or pollen, you can imagine there's going to be major problems with these. So they essentially, if it's for humans in particular, and this, say C became the primary chromosome in an oocyte, that's effectively a monosomy, right? I mean, if you've lost that much of the chromosome, all this part down here, you might as well think of that as a monosomy, and it's probably going to be lethal. Even if you just lose a little piece down here, you still could have issues with it and be problems. Now, traditionally, I've shown it a little more like this, okay, kind of the same idea. But in our case, we have what's called a dicentric chromosome. Again, that's the one they're showing breaking. And we have this acentric. And again, with things that don't have centromeres tend to be lost, particularly in this process. So even if it wanders over and gets into the oocyte, it's not going to be recognized as a full chromosome. And even if it does, it's going to cause some issues because obviously it doesn't have A and B and centromeres and things that it needs in it, right? Well, I would practice with this a little bit, okay, working from this and kind of playing with it. Now, one of the things I want to warn you about is that when students do this, they tend to always want to loop it from the bottom, right? What I mean by that is if you're practicing with it, you don't have to take the bottom one and be the one that loops it, right? Like that. I can't draw very well with this thing, but it doesn't, it could be the top one that loops, okay? So be prepared to think about it in terms of the fact that it could come along and do this, okay? And then the bottom one be the one that's like that. It doesn't really matter which chromatids do the curling. It's this one or this one, but you could get different outcomes if you do that. So try that out. Try it out with a pericentric. Okay, try it out with a paracentric, and just see and just play with it a little bit and see what you end up with. See if you get these outcomes that you see here. This is kind of important to work through it. All right. Okay. Now let's move on to the other ones, which are translocations. So with inversions, we didn't lose or gain any chromosome material as the inversion occurred. You might lose or gain some when you go through the meiotic process later, but as far as just the actual action of it, the chromosome remained all the DNA that was there when you started. Translocations, that doesn't necessarily have to happen. Translocations are an exchange between non-homologous chromosomes. So unlike we would have with recombination and crossover, now we've got a chromosome pair off down here somewhere, and somehow we're a chromosome down here, and somehow we're exchanging information with it. And this sort of shows that we have A, B, C, D, E, F, G, our standard chromosome, completely different chromosome. This might be chromosome number 2, and this number 14 or something like that. And somehow pieces of 14 end up stuck on the end of 2, and 2 ends up stuck on the end of 14. This is known as a reciprocal translocation. A reciprocal translocation is where part of the first chromosome and part of the second chromosome that we're working with, these non-homologous chromosomes, actually exchange material. So the one you're looking at here would be called a reciprocal translocation because they reciprocated, right? One chromosome gave up some part, but picked up another part. The other one picked up a part and gave up a part. Okay? They can be non-reciprocal. Non-reciprocal would mean that you simply come along and they D and E ends up on the bottom over here of this one, okay? But you just lose it. Now, genome-wise, you didn't lose all the material, but you might have lost a big chunk of this chromosome here over here. So reciprocal, they both exchange some information. Non-reciprocal, the DNA just moves from one spot to another. Now, there are also things known as Robustonian translocations. Robustonian translocations are where are called centric fusions. And these are where, because the centromere is really dense, and so what will happen sometimes is those centromeres will overlap like this, and they're sticky and they'll form a kind of a larger centromere where you've got most of this one chromosome and most of this one stuck together. And you end up with the centromeres being over here and sort of this modified chromosome that we would see here, right? That's happened quite frequently in systems. We know that's happened, and it's important in speciation events. And it tends to occur in things where you have like these little satellite chromosomes. Like in humans, we have 21, 22 down here. The centromeres are way near the top. You have very little DNA above that, and they might fuse up with, say, 15 right here and join up to form at the centromeres. So you'd lose a little bit of material maybe off the top of each one of these, but the majority of the chromosomes would still be there. Those are known as centric fusions or Robustonian translocations. And we know they're important in speciation events. I've been working with a group that have been looking at these shrews, the southern short-tailed shrew, looks something like this, little guy found commonly in the southeastern U.S. And these little shrews, really interesting that within a very short distance, they can have major changes in the chromosome structure due to these fusion events. And that's kind of what's showing you here. In case some of them have this fused chromosome here, which is a fusion of these two. This one was a fusion of this one, and this one makes this one. And they have different combinations of those in different populations, and they don't have to be very far apart. For the most part, they're 2n46, not the same number that we see in humans. Of course, they're different chromosome structures. But if you go on the bluffs, the Tennessee River or the Mississippi River in, say, Tennessee, if you go somewhere, if you've ever been to Memphis or you go anywhere up in that area, you know that the city is built on a bluff, and that a bluff extends north and south of the city, and it represents the easternmost place that the Mississippi River has wandered over in its long history. So this is the river is at its easternmost point, and it's wandered back and forth across the Mississippi Delta for millennia, going all the way over to Arkansas. If any of you from Arkansas know about Crawley's Ridge, which is a number of miles. It's really flat, and all of a sudden you go up this little ridge. Well, that's the western extent to where the river has meandered back and forth. Well, anyway, along that ridge, there are populations. There are populations of brina in all sorts of places, but these have been studied north of Memphis, and within a very small area, you can actually see chromosomal variation that's dramatically different. Even sometimes within just, meters of each other almost. You can see 2n numbers all the way as low as 34. That's where you've got lots of these fusion events going on. You can have 40 anywhere in there, 34 to 40 within a given population, with the real number, the common number being 46. And it looks like this is incipient speciation because if you fused a bunch of these together, it's highly unlikely that you're going to effectively. It's going to be like trying to put the horse and the donkey together, right? You may be able to breed, but you're probably going to end up with sterile situation. So it's probably leading at some level to reproductive isolation, which is one of the driving forces of speciation. So the question is then, why is all this going on? What's happening? No one really knows yet, but there's some nice studies going on trying to figure out what's really happening in these populations and what's driving the system. All right, now let's take it back to humans, and this applies to horses and dogs and all sorts of things, but we're going to use an example again from humans. This is, we're going to talk about translocations and the effects that it can have, okay? So let's take a situation where we've got normal 21st chromosomes, right, pair, those small chromosomes, and normal 14th. Well, there's a fairly, I wouldn't call it common condition, but there is a condition in humans that reappears where much of the 21st fuses to the 14th. Okay, so remember the 21st is a small chromosome. There's also the same thing with the 18th chromosome and a couple of the others. This is not unusual, but in this particular case, we get an outcome that you'll recognize. Okay, so we have a normal individual here, and then we have what is called a translocation carrier. That individual has a 1/21st, one normal 14th, and then this hybrid 14 that's been translocated with a 21st. And there's so much of the 21st here that this is a normal individual, okay? It's not a monosomy because even though it's missing this chromosome 21st, it's enough of it here that work. And they'll actually go through meiosis and connect. And it's in that case, you've got 4 things trying to interconnect and to make the system, and it's very complicated. Now, when they go through meiosis, that's where you end up with interesting issues, certainly with fertilization. If a normal individual goes through meiosis, it's going to produce a gamete that's got 1/21st and 1/14th. But look what's going to happen up here. Okay? Sometimes we're going to get this 14th in a normal chromosome. Sometimes we're going to, or this 21st, excuse me, and a normal 14th. Sometimes we get the 21st and a hybrid. Sometimes we get this missing part here and a hybrid, and sometimes we get the missing part and a normal. So we can get at least 4 common outcomes. One, it could be a completely normal gamete. One can produce a gamete that gets this null here. Didn't get this, but he got a 14th. Looks like that. You can get the 21st in this hybrid, like that. And then finally, you could get the null and the hybrid, right? Those are this joined chromosome right here, okay? Now, when you get that, of course, everything's fine. They're gametes, right? Until you combine them with, say this is the egg over here, and these are the possible sperm, or vice versa. If you fuse these two together, I think you can see you're going to end up with a 21st and a 21st and a 14th and a 14th. That's going to look fine. Okay, that's all going to work out okay. All right, so what I've done here is I've just taken my 21st from this gamete and put them all down here. I've taken my 14th and put them that. So this part is just from here, okay? It's absolutely normal, okay? We've got no problem. Now we're going to start dropping these down. Well, I'm going to drop down a 21st. I'm going to drop down a 14th. And when I do, totally normal human, 46 chromosomes, everything's paired up, got homologs or everything else, there's no problem, okay? All right, but let's drop the second one down. All right, now what do we have? We've got 2 normal 14ths and a 21st, but we're missing something right here. This would be called a monosomy, and generally monosomies are lethals, right? So these would be early lethals, right? Then we drop the next one down. Well, the next one has a 21st, a 21st, a 14th, a 14th. Oh, wait, there's another part of a 21st here, okay? All right, well, that's enough of that 21st to function. This individual functions, so that must mean we have essentially 3 21st. And what we called that before was a trisomy, and we know that the trisomy 21 leads to Down syndrome, right? So these are called Downs individuals, and in fact, they are Downs individuals. And then finally out here, we have this hybrid chromosome, and you call it hybrid, but it's a complex chromosome with a 14/21st translocation. That's another carrier, all right? Okay, and here's an example of what it really looks like. Here's a 21st, 21st. Here's the 21st, almost all of it connected into the 14th right here, and then here's a normal 14th here. So this would be a Downs individual, okay? All right, this is what is called familial Downs, right? Familial Downs. We mentioned early on when we talked about aneuploidies with humans, and we said 95% of aneuploidies are spontaneous. They're de novo. They're not in any family. All of a sudden you have a Down's child or you see something else. 5% or so turn out to be familial, meaning that if you took the pedigree back far enough, you'd see that there was Downs here and then every now and then. The reason for that, and when you do also look in those pedigrees, there tend to be a lot of miscarriages because you get a lot of this stuff going on, right? That's because you've got a translocation going on in there. There's also translocations for Edwards. There's translocations for anything. There's known translocations in horses, all sorts of things that you can actually see these carriers in production. But notice the carriers continue down. They're normal. you get some normal possible progeny, but you can also get this reoccurring Down syndrome. Now your book shows it slightly differently, like this, translocation carrier, or something like that. It doesn't really matter. Here's the Downs. They show that you can get a 14th thing. It doesn't really matter. You see the point I'm making is that you have these translocations. And they don't have to be, all the way leading to Downs. You can get other things. One of the areas that we're starting to see evidence of translocations having effects are in some types of tumors. This is just an example of some T-cell lymphomas. And note, these are different tumor types. And you'll notice these parts over here are telling you which ones are interconnected. So this is a translocation from the first to the second, and that's this right here. So here's the first chromosome, and here's the translocation onto the second right here. There's a 12-15. So if you go down and look at 12 right here, and you look over at 15, there's been a translocation there. There's translocations, and some of them from 14 to 3. Okay, so part 3 is jumped down here on 14, and that's showing an example of that. These are all lymphomas. They're all different variants of that lymphoma, and they all have these translocations going on. It's one of the problems with trying to develop ways of attacking tumors and cancers is that even though it's, we categorize it as a lymphoma or as a so whatever carcinoma it might be, a melanoma, there's different forms, different things cause it. So going after one where it's resulting from a 1-2 translocation, when in fact it is something like a 14, 12 translocation down here, can have very different, effect. Trying to go after that one, they may in fact be more aggressive or less aggressive. They may have different forms. So just trying to go after them is problematic, and just identifying the underlying genetic cause can be problematic in these. So, but in this case, we can see really clear evidence that there's been translocations that are at least associated with the tumor. Now, whether they cause a tumor is a whole other question, but they certainly are associated with the tumors. All right, well, that concludes everything we're going to talk about as far as chromosome mutations. We started out talking about euploidies and these things we call polyploids. We talked about autopolyploids and allopolyploids and all the euploidies. We gave some examples of aneuploidies and various forms of those. We didn't mention nullisomy. Nullisomy is when you lose both homologs. We didn't mention it because it's lethal in humans. There are no good examples of it. There are some been argued again for tumors, but it turns out even in those cases, very often, a lot of the DNA is actually just translocated. So while the chromosomes aren't there, the DNA just moves somewhere else. And so it's not a true nullisomy in that sense. But we did talk about some of these others, monosomy being a Turner syndrome. So now we've gone through all the rearrangements, duplications, deletions, paracentric, pericentric. You should be able to kind of draw any one of these, using some outcome if you want. So besides just memorizing what they associate with, be able to look at them and see, oh, I know what that is. That's a trisomy. Oh, that's a pericentric inversion or something. Okay. All right. Well, that's a long way through the chromosomes, but that's one of the unique material for any introductory genetics course, so it's worth the time to provide you with a lot of information on it, and I think it's all kind of important, and hopefully some of it's interesting to you. All right, so now we're going to move away from that, and we're going to move back into the molecular genetics side and talk about some of the ways that what genes are, and then how to transcribe those genes and how to regulate those genes.